So we are live.
Okay.
Assalamualaikum and a very good evening to all our viewers who have joined with us tonight. I hope everyone is doing great. I'm Dordana Hussein Priya, the Deputy Director of International Relations at Community of Biotechnology, your host tonight. I welcome you all to the CRISPR-Cas9 2020 organized by the Community of Biotechnology, abbreviated as COB. Community of Biotechnology is a national non-profit organization in Bangladesh founded in May 2017. The organization works with students, researchers in enhancing their expertise and future scopes in their respective fields of biology. The community also involves the assessment of individuals from both public and private universities of the country and offers hands-on training sessions. We frequently conduct seminars, webinars, workshops in order to help the students who are interested in this particular field. It gives me immense pleasure to announce that the CRISPR event has reached out to almost 100K members, and we have been successful in connecting with almost about 70 plus countries. Throughout our journey, we have had Ruin Kamens, who is the Executive Director of AdGene. Then we have Kate Adamella, Zeba Islam Shiraj ma'am, Tofazil sir, Zakir Asir sir as our advisors. So coming to today's session, I'm honored and thrilled to welcome our special guest speaker, Dr. Matthew Porteus. So to say the least about him, Dr. Matthew Porteus is a professor of Pediatrics, Department of Pediatrics, Divisions of Hematology, Oncology, and Human Gene Therapy at Stanford School of Medicine. His research is focused on developing homologous recombination-based therapies for genetic and other diseases. He has a clinical practice at the Lucille Packard Children's Hospital, where he is an attending physician for the pediatric bone marrow transplant service. Dr. Matthew Porteus graduated Magna Cum Laude from Harvard University before completing his MD and PhD degrees at Stanford. His PhD mainly focused on the molecular biology of mammalian forebrain development. He then completed his residency training in pediatrics at Boston Children's Hospital and his fellowship training in pediatric hematology oncology at Boston Children's Hospital and the Dana-Farber Cancer Institute. So moving forward, today's session is on CRISPR-Cas9 genome editing to create transformative medicines, one of the hottest topics nowadays, and probably there is no one suited to talk about this than my guest, Dr. Matthew Porteus. So without delaying any further, I would like to request Dr. Matthew Porteus to start one of the most awaiting sessions of the event. So yeah, please go ahead. The stage is all yours now. Thank you. Best of luck.
Thank you very much for that very kind. Thank you very much for that very kind introduction, overly generous, and thank you all for joining me in this final talk at CRISPR Fest 2020. It sounds like it's been an incredibly exciting and engaging conference, and congratulations for the community of biotechnology for putting on such an excellent and timely conference. So I'm a firm believer that new technologies will allow us to improve the health of people all over the world. And this has been demonstrated through the history of medicine, with each technology starting out with simple sanitation, and then the development of antisepsis and anesthesia, through the development of vaccines and small molecules, and then over the last several decades, the development of what we call biologics, have all resulted in the improvements of human health, because one platform might not be sufficient to treat everything. That is not to say that, for example, small molecule drugs are not effective. They're highly effective. It's just that there are certain diseases for which they are ineffective. I do believe we are at the dawn of a new era in which we will use so-called living drugs, drugs that that are inert and have simple chemistry half-lives, but instead we'll be using cells and genes which have entirely different pharmacokinetics and pharmacodynamics. And particularly if you think about stem cell-based drugs, which I'll be talking about more in the rest of the talk, these have the potential to have lifetime durability. So the possibility that we could use a single dose of stem cells to cure a patient definitively for their lifetime. Now, we are just at the dawn of this era, and while there's exciting early demonstrations of this, I think, in the next 5, 10, 20, even 30 years, we're going to see an explosion in these types of drugs. So specifically, I've been working on the idea of engineering cells using genome editing. And the reason I think many of us are excited about genome editing is its incredible precision. So you can modify the genome with single nucleotide specificity. And the most well-developed form of genome editing is based on the idea of designing a protein, what we call nuclease, that is engineered to recognize a specific DNA sequence. And when it binds that sequence, it makes a break in the DNA, so-called DNA double-stranded break. Now, DNA double-stranded breaks are something that occur tens of times per day in every cell in the body. And so our cells have developed redundant repair mechanisms to fix these breaks. And the two major ways that cells fix breaks are by non-homologous end joining, which is a complex of proteins that essentially stitch the two ends back together. And this is actually generally a pretty accurate process. Our estimates is that 7 to 9 times out of 10, it puts the ends back together in a precise way. But occasionally, it will create little small insertions and deletions at the site of the break in the process of fixing it. And so this now allows scientists and researchers to create specific mutations at specific sites in the genome. And this could be in the coding regions of genes, it could be in regulatory elements, and it's become an incredibly powerful research tool and is also being applied to develop new medicines for patients as well. But for most of the diseases that I'm interested in, which are caused by mutations in a single gene, the idea of creating a new mutation in that gene doesn't seem to work. What we really have to do is be able to use the homologous recombination pathway that cells also have to fix breaks. And in the recombination pathway, again, a larger complex of proteins than the non-homologous end joining pathway, functionally identifies a undamaged piece of DNA that has a sequence identity to the site of the break, and then functionally makes a copy of that undamaged DNA and pastes it over the break. So one can think of it as a copy and paste mechanism. And the advantage of harnessing this recombination pathway of repair is that you can do a number of different things to the genome with single nucleotide precision. You can make single nucleotide changes in the genome. You can introduce a cassette of a gene and put it back into its own gene. You can identify what we call a safe harbor, a location in the genome that we believe if you insert a promoter in a new gene or a new set of genes, that it won't cause any deleterious effects. And then you can start to build even more sophisticated systems where you take one gene and you put it into another gene. And so you repurpose the way the cell is working by doing this. And so I will most spend time doing sort of the first set, which is to look at single nucleotide precision. So now the field of genome editing has been around for several decades, but has really been transformed in the last half decade by the discovery and the development of the CRISPR-Cas9 system. But I will point out that there are at least 3 different other platforms that allow one to make a site-specific double-stranded break in the genome. And these include homing endonucleases, the first class of nucleases that were used in genome editing, zinc finger nucleases, and tau effector nucleases. Now, all 3 of these earlier generation nucleases were based on having a protein sequence recognize a DNA sequence. And so there were significant challenges in cracking this protein DNA code, some of which have been solved, but many of which remain. But what really, in 2013, when the CRISPR-Cas9 system was discovered as something that could be used first in a test tube and then in cells, it completely changed things, because instead of having to crack the protein DNA recognition code, it harnessed the well-known Watson-Crick base pairing code, whereby simply a guide RNA complex to a Cas9 protein could recognize the target sequence through Watson-Crick base pairing between the guide RNA stretch and the DNA, the genomic DNA sequence, and that would bring the Cas9 protein to the specific site that we wanted. And the code, of course, Watson-Crick code is, of course, well known. Now, I do think it's worth calling out the amazing where the CRISPR-Cas9 system came from, which I think is an incredible story in and of itself. So as we all know, humans and we have an adaptive immune system, an immune system that allows us to react to foreign infections. And we know that if we don't have pre-existing immunity to a new infection, the devastating consequences that can have as we're living through this SARS-CoV-2 pandemic. But I think it was really surprising to people that bacteria have an adaptive immune system. But if you take a step back, you realize, well, bacteria also can be infected by phage, the viruses for bacteria, or by plasmid DNA. And a number of different researchers, including Emmanuelle Charpentier, who won the Nobel Prize this year, were trying to understand the genetic and biochemical mechanisms of this bacterial adaptive immunity. And it was from this bacterial adaptive immune system that our CRISPR-Cas9 genome editing system came from. And it was really this paper in 2012 from Emmanuelle and Jennifer Doudna, who with Martin Ginnick, who demonstrated that they could pull out the CRISPR-Cas9 system in bacteria into a test tube and reduce it to two components: the Cas9 protein, which is a multifunctional protein that unwinds DNA and cuts DNA, and a single guide RNA that complex to the multifunctional Cas9 protein and guide the Cas9 to where it should make that double-stranded break. And of course, we're all excited about the awarding of the Nobel Prize in Chemistry for the discovery of this two-component system. Now, so with that in mind, we were using zinc finger nucleases and talons, and then the CRISPR-Cas9 system to try to modify human stem cells as a way of potentially developing new therapies. And what we learned, unfortunately, as did many others, is that when we deliver naked DNA plasmid molecules, sort of the usual molecular biology reagents we use to deliver the Cas9 protein into cell lines like HEK293 T cells or HeLa cells or K562 cells, where the system worked wonderfully, we found that when we delivered these same naked DNA molecules into human stem cells, we activated a type 1 interferon response, a toxic type 1 interferon response. And again, stepping back, what you realize is, well, of course that's what's going on, because naked DNA should not be in the cytoplasm. And if it is in the cytoplasm, it could be a sign that the cell is being infected by a virus. And what do cells do when they're infected by a virus? Well, they activate the type 1 interferon response. So we and others have spent many years trying to figure out how to deliver these very powerful genome editing reagents into human stem cells without activating this type 1 interferon response. And the system that we have come up with is an ex vivo system in which we purify our cell of interest. In this case, I'm showing a micrograph of hematopoietic stem cells. We put them into we optimize our cell culture conditions so that they are healthy and dividing, because the recombination pathway is most active, is really only active in the S and G2 stage of the cell cycle. We then take the CRISPR-Cas9 system, and instead of delivering it on a DNA expression plasmid, we actually deliver it directly as a fully formed ribonuclear protein complex, where the Cas9 is purified and is complex to a synthetic guide molecule that has been modified at the ends to protect it from degradation. And we deliver this RNP into our cells by electroporation, by shocking the cells, creating holes in the membrane, and allowing the RNP to enter the cell, and then through the nuclear localization signal on the Cas9, enter the nucleus. So that gets the nuclease into the nucleus of these cells without activating the type 1 interferon response. But we need to also deliver the donor template, which needs to be a DNA molecule. And the DNA molecule, as I said, is what is sensed in the cytoplasm. So for that, we and others have harnessed a non-pathogenic virus that has evolved to deliver its DNA content to the nucleus of cells without being detected. This is recombinant adeno-associated virus serotype 6. And we have gutted the normal genome of AAV6 and replaced it with our donor template. And so we purify our cells, we electroporate the RNP, and then we add the AAV immediately after the RNP, and that allows us to get those to activate the homologous recombination pathway. So with that system in mind, we can think about applying that in many different ways, but here are two broad categories. One is to apply it to stem cells and rescue a missing function. For example, a hematopoietic stem cell from a patient with a genetic disease, one could try to correct that mutation. And that's what I'm going to focus on. But an even broader pathway is to use the precision of genome editing to add new functions and potentially new therapeutic functions to these stem cells or other cells. So in a way, combining the precision of genome editing with the broad possibilities of synthetic biology. So I'm going to focus on sickle cell disease. Sickle cell disease is I think the most common serious genetic disease in the world. Every patient has the same mutation in codon 6 of the beta globin gene. A adenine is converted to a thymidine. This leads to a single amino acid change where a charged amino acid, glutamic acid, is converted to a polar amino acid, valine. This polar amino acid in the beta globin protein creates a short hydrophobic surface that is exposed when the hemoglobin molecule unloads its oxygen. And this short hydrophobic surface, when it's exposed, can interact through hydrophobic van der Waals interactions to create a polymer of the hemoglobin molecule. And so instead of the hemoglobin tetramer being free-floating, we now create these polymers, and it converts these round red blood cells to sickle-shaped red blood cells, giving the disease its name. And these stiff sickle red blood cells can form occlusions in the small blood vessels of the body, preventing the healthy red blood cells from delivering oxygen to the distal tissues. So that can lead to strokes in the brain. It can lead to something we call acute chest syndrome in the lungs. It can lead to deep, painful bony crises in the bones. And it basically results in the deterioration of all of the organ systems of the body over time. Now, this is a disease in which approximately 300,000 people are born per year, 80% of them in sub-Saharan Africa. But it is still a disease that is present throughout the world. And in the United States, the median lifespan is in the mid-40s, although in other parts of the world it is much shorter than that. And of course, sickle cell disease represents a class of diseases, these so-called monogenic diseases, and there's estimated to be perhaps 10,000 such diseases affecting 350 million people around the world. So perhaps between 5 and 10% of the people around the world have one of these diseases. Now, most of these are quite rare, but every patient with a genetic disease, of course, affects a much larger community of people, immediate family, the communities around them. And so one can imagine that if one could cure a patient with a serious genetic disease, you're not only helping the patient, but you're helping the entire community. So what I've described to you is that the disease is caused by dysfunctional red blood cells. Now, red blood cells only have a short only have a short lifespan. In non-sickling red people, the lifespan is about 120 days, but in sickle cell disease patients, the lifespan is only about 10 days. So if we want to get a durable lifetime cure, we don't want to focus on fixing the red blood cell. Instead, what we have to do is try to fix the hematopoietic stem cell, which is the cell that would give rise to red blood cells and actually all the other blood cells of the hematopoietic system for the lifetime of the patient. And so one of the ways that we can give so-called corrected hematopoietic stem cells is through allogeneic hematopoietic stem cell transplantation. And in hematopoietic stem cell transplantation, we identify a person who doesn't have the disease but is an immune match to the patient. We then give the patient high doses of chemotherapy to ablate their hematopoietic and immune systems, and then transplant. And by transplant, we actually are able to give these cells simply as a blood as an infusion through an IV. And these hematopoietic stem cells can, from the donor, can migrate to the bone marrow of the patient and start making a new blood system. And this was first used to cure somebody of sickle cell disease in 1984. And it was a single patient. It was a young girl who had AML, a blood cancer. And to treat her blood cancer, she received a bone marrow transplant, hematopoietic stem cell transplant from her brother, who was an immune match. And her brother had sickle cell trait. And what you can see is that she had almost all hemoglobin S before the transplant. And after the transplant, she had 40% hemoglobin S, which was the same amount as her brother. And what we know is this ratio of 60% hemoglobin A to 40% hemoglobin S causes sickle cell trait, which is generally a healthy condition. It's not even a disease. And this has now been applied much more broadly. And 4 years ago, a consortium of investigators reported on the outcomes of bone marrow transplants, hematopoietic stem cell transplants using matched sibling donors, and found that the overall cure rate was excellent, around 95% or above. The challenge is only a small number of patients actually have these donors, and there are still some immune complications. So while hematopoietic stem cell transplant proves that one can use corrected hematopoietic stem cells to cure the disease, it's not accessible to the number of patients we would like. So what that leads us to is, well, instead of using someone else's stem cells, can we use gene editing to correct the patient's own stem cells? And because they're their own stem cells, the issue of donor availability goes away because every patient can be their own donor, and the issue of the immune reaction goes away because they're their own cells. So a large team of people that I've been working with over the last number of years have been working to apply the genome editing system I described to you to sickle cell disease. And when we take CD34 cells, which are CD34 is a cell surface marker of hematopoietic stem cells, and we correct them using the genome editing system I described, we can get in the laboratory between 60 and 80% of the alleles corrected. So an excellent number. So after we take these cells through this relatively complicated process of purifying them, putting them in the cycle by culturing them and delivering the RMP and the virus, do they still maintain the properties that we need them to maintain? And namely, will they make red blood cells that don't have sickle hemoglobin? And do they maintain their stem cell properties that allow us to think that they might last the lifetime in a patient? So when to answer the first question, we look at what kind of hemoglobin do these corrected stem cells make? And we find that after correction, we convert them from cells that make essentially all hemoglobin S, the pathologic hemoglobin, to essentially all hemoglobin A, so 90% hemoglobin A. Mentioned to you before that we think even 50% or 60% hemoglobin A is sufficient, so we've certainly exceeded that threshold. Now, the question of whether they retain their stem cell potential is a really complicated one, because we are modifying human cells, and the only way to really test whether they are true stem cells is to put them back into another human. But before we're allowed to do that, we have to have some evidence that they do retain those properties. And so the best available model now is to take human cells and transplant them into immunodeficient mice, and then see what sort of level of engraftment do our gene-corrected cells achieve in these immunodeficient mouse models. And what we find is encouraging but a bit confusing, because depending on where we get our hematopoietic stem cells, whether we get them from the peripheral blood from sickle cell disease, where we get them from the peripheral blood of patients with sickle cell disease who've been mobilized with a drug, or we get them from healthy volunteers, we see the level of engraftment changes. So in some senses, our gene-corrected cells engraft very well. From healthy donors, they don't engraft quite as well, where we only get about 20% median correction frequency. But in the sickle cell disease patients, we get about 30% engraftment of the gene-corrected cells. Nonetheless, depending on who you talk to, the bar for potentially curing or significantly changing the course of the disease is between 2 and 5% and 15%. So we've exceeded those numbers. So this leads us to think that we've done a whole host of safety studies, which I'm not going to go into, and we've done a whole host of studies taking the process in the lab and moving it into a large GMP manufacturing facility, allowing us to submit an application, what we call an IND, to the US FDA to ask permission to start a clinical trial in which we would identify a patient with severe sickle cell disease. In this case, I'm showing a child. We would start with adults first. We would mobilize their hematopoietic stem cells from the bone marrow into the periphery, and then use pheresis to purify those cells, or to harvest those cells and then purify them on a Miltenyi Clinimax. I'm showing a prodigy here. And then after purification, the cells would be brought into this GMP facility, where they'd be manufactured in the way I described, by culturing and then delivering the Cas9, which we get from Aldevron, a guide RNA, which we have from Agilent, and using AAV6 that's made by a contract manufacturing organization, and using an electroporation system from Lonza. And after the correction of these cells, we will test them to make sure that there's a sufficient number with a significant allele correction frequency. And if they pass their tests, then the patient would come back into the hospital and receive an autologous bone marrow transplant. And we're optimistic that we might treat our first patient in the early part of 2021. I do want to point out that while I've summarized where we are in 2020, if you trace back how long people have been working on this, I trace it back at least until 1985, when Oliver Smithies described that he could modify the human beta globin locus in a cancer cell line at a frequency of 1 in a million cells. And then we had Maria Jason showing that homing endonucleases could increase that targeted integration frequency in general to 0.5%. And so it was not a process that occurred overnight, but instead has been a process that occurred by the iterative, slow developments and discoveries from people from around the world, and then applying those in a very focused and concerted manner to this disease. Now, in the last couple minutes, I just want to take discuss two other topics as it relates broadly to the application of human genome editing. And the first, as the CRISPR-Cas9 genome editing system became recognized as being so powerful, there has been lots of discussion about how should this system be applied, and specifically, should it be applied not just to somatic cells to cure disease, as I've discussed, but what about the possibility of using the system to do human embryo editing, which would result perhaps in the prevention of people being born with genetic diseases, but would certainly result in humans intervening in the natural transmission of genetic material from one generation to the next, a possibility that many people find, many people have thought is a line that should not be crossed. So this committee was formed, and I was fortunate enough to be a part of it. And the committee actually said that it was a line that could be crossed, but only if a number of very strict criteria were met. And I will say that when we released the report several years ago, none of these criteria had been met. And several years later, I can say that none of these criteria have been met. And so while it's not a call for a moratorium, the reality is it's a functional moratorium. And the criteria span not just include technical criteria, but also include governance and input criteria. But I want to just focus on two of the really strict technical criteria. The first one is that one would not do this if there's an alternative way of achieving a child who's genetically related without doing editing. If you don't need to do it, why cross the boundary? And while in vitro fertilization with pre-implantation genetic diagnosis is not widely available and is not incredibly efficient, the reality is for most couples who are at risk of having a child with a genetic disease, this is a process by which they could have healthy children without a genetic disease. And so there would be no need to have humans intervene in the natural transmission of genetic material across generations. The other really strict criteria is the committee said that you can only edit sequences to sequences that are already common and known to be healthy in the human population. That is, we're not going to create new sequences that aren't already present in humans. Now, a definition of common and known to be healthy can be of some discussion, but I think this really is a pretty strict criteria. I will point out that when I discussed my sickle cell editing program, we actually were not only reverting the variant that causes the disease, we're also introducing other sequence changes into the genome to help facilitate the gene correction process. And per this criteria, the process I described in somatic cells would not be permitted in embryos because it introduces DNA sequences that are not present in the human population. And then the last issue I want to discuss, and I think it's probably something, I guess I'm a little embarrassed to discuss it with this audience because you are all too aware of it, is the issue of equity and distribution. So I am very fortunate to work at Stanford University, where I have a very fantastic infrastructure and support to do the research we do. I get to work at a state-of-the-art children's hospital. And of course, academically, I'm a success if we're able to treat even a single patient here. But as I discussed, most of the patients with these genetic diseases don't live in Palo Alto, California, but live all around the world. And while the distribution of advanced medicines from to patients all around the world is not a problem or challenge that's restricted to genome editing, I actually think because genome editing is such a new field, we might have the opportunity to solve this problem before the therapies get locked in and not become available. I hope I'm making myself clear that the field is so new, we have the opportunity to figure out ways to get these therapies to all parts of the world rather than just have them become available to a small number of people who by chance might have been born in one country and not another. So with that, again, I want to thank you for the invitation to speak at this exciting CRISPR Fest 2020. I have a fantastic, to take what we're doing requires collaboration among a large number of people, including people in my own lab, funding from a large number of sources, collaborators in our GMP facility to help us take what we do in the lab and make the process compatible with actually giving to patients, clinical collaborators, and then collaborators at Stanford and other institutions and collaborators in biotech and pharma. The genome editing field requires these sort of collaboration. It's been very exciting. So once again, thank you, and I look forward to answering any of your questions, or all of it. Well, I might not get to all of your questions, but many of your questions. Again, thank you.
Thank you so much, Dr. Porteus, for this wonderful session. It was a great talk. I personally learned a lot from this session. So thank you so much. Now let's move forward to the question and answer segment of the event. So can we have the questions, please?
Thank you, Ashik. Thank you. I'm going to apologize in advance if I don't say all your names correctly, but I will try my best.
You're fine. It's completely fine.
Oh my goodness. Thank you so much.
It's great. It feels great to have you, actually.
Yeah,
That's why.
Man, I'm walking on air right now. This is very, very nice.
Okay, so we have a question.
Yeah. Yeah. So that's an excellent question. And so the way the system works is that if a bacteriophage or a piece of DNA enters the bacteria, the broader CRISPR system chops up that incoming DNA into small pieces and then takes the small pieces and integrates it into the bacterial genome. And it's these integrated pieces of DNA that actually give the system its name, which really stands for clustered, regularly interspersed palindromic repeats. Whew, got that all out. Then what happens is the bacteria expresses small RNA from the little fragments of the bacteriophage, and those little small RNAs, which now we call in this case is called we call them guide RNAs, is complex to the Cas9 protein. So the next time the bacteriophage or DNA comes in, there's a nuclease that is ready to sense it and chop it up. So that it's true the bacteria sort of have to survive that first infection, but once it does, it then is able to chop up the incoming DNA. And of course, that's very similar to our adaptive immune system, where we get infected with a virus or a bacteria. And the first time we get infected, we have to be able to survive that infection. But then after we survive it, we have an immune system that's ready to prevent it from causing more harm the second time, which is, again, why vaccines are so important, because it allows us to develop that immunity before we even get that first infection. Thank you.
Thank you for answering. The next question is from Tayyab Moin.
Yeah, so Tayyab is asking, okay, it looks great for sickle cell disease. How about for autoimmune diseases or cancer mutations? And I think, one day we hope that we'll be able to do this. I think autoimmune diseases, we can imagine doing this in a couple of ways. One is that there's a small number of patients who have genetic autoimmune diseases. My colleagues work with a disease called IPEX, which is caused by mutations in FOXP3. So one could imagine correcting that mutation. But most patients don't have a genetic or a single gene cause of their autoimmune disease. So I think for autoimmune diseases, we're going to probably have to harness what I didn't talk too much about, which is synthetic biology, to re-engineer the immune system in a way that suppresses the autoimmune or autoreactive T cells. So I think it's going to be possible, but it's going to take smart immunologists. In terms of cancer, I don't think we're ever going to be able to take in vivo edit the cancer-causing mutations in all the cancer cells. So again, I think what we're going to have to do is use genome editing to create immune systems to eliminate cancer. So harness the natural ability of our immune systems to eliminate cancer that have failed, and that's why cancer developed, but then use genome editing to give those immune systems more power. And it's really building on the CAR-T transformation that's occurred. So again, thank you, Tayyab.
Thank you. Next question is from Tanji Mishra
Yeah.
Rahman.
So how to target different transcriptional variants with CRISPR-Cas9. Yeah, that's something we've been trying to, with my colleague Rosa Paqueta with IPAC and FOXP3, is that there are different splice forms. And so this is where you can have a sort of one size fits all, but in the end, you have to know your gene really well. So, for example, sometimes we start by trying to target exon 1 where the start codon is, and we learn that, that doesn't capture these transcriptional variants. So we have to move our targeting site to account for these transcriptional variants. What I would say is I don't have a one-size-fits-all answer, except to say that it still requires deep knowledge of every gene to do this the right way. Yeah.
Okay. Next question is from Tayab Moin again.
Thank you. Yeah. Obviously, I'm an optimist, and I believe that autologous stem cell transplantation using genome editing should be the way that we cost effectively treat genetic autoimmune diseases. And we didn't get in, I mentioned the challenge of making this cost effective, but I truly believe that what we can do now is convert the powerful biology that we now understand and use engineering to make our reagents cheaper, to make the cell manufacturing more automated. And so that one day, instead of having a process that costs, hundreds of thousands of dollars, just have a process that might only cost, thousands of dollars. And I truly believe that this is possible. And once we get through our sickle cell clinical trial, I can tell you it's going to be one of my major interests in trying to make this more cost effective.
Next is from Andali Popu.
Yeah, that's a great, very practical question. So there's a lot of programs out there that try to allow you to score for which guide RNA might be more active than another guide RNA. In our hands, those scoring programs, they're not wrong, but they don't help us. So what we generally do is if we say we want to target this 50 base pair region of the genome, what we generally do is just make 3 to 5 guide RNAs in that region and then just test for the best one. We do use the bioinformatics off-target programs as a way of eliminating guide RNAs that look really bad, that might have a lot of off-targets. So we use bioinformatics to say, okay, this looks pretty specific, and then we use experiments to pick out which of the 3 to 5 really is the best. And we find that about 50% of the guide RNAs that we pick work great, and about a quarter work okay, and about a quarter, for reasons we don't understand, just don't work at all.
Okay. Next is from Tayab Moin.
Yeah, Tayab, this is you're spot on. So let me just step back a bit and give some background about why your question is so important. So when we do currently when we do an autologous stem cell transplant, we still have to give chemotherapy to get rid of the stem cells that are sitting in the bone marrow. And that chemotherapy, while effective, has toxicities, both short and long term. And those toxicities are significant enough that it's a barrier to being able to do this broadly. And so what people are working on now is, can we replace either radiation or chemotherapy with biologic agents to create space in the bone marrow? And these include antibodies to create space, and some of my colleagues at Stanford are developing this. It could be even an engineered T cell to create space. We will see. But I'm very optimistic that we will have these non-toxic biologic agents that will create niche space and thereby also make the process much safer and much more available. I think our goal is the current autologous transplantation regimen usually involves being somewhere between 30 and 60 days in the hospital. What we want to do is turn this into something that's completely outpatient. Now, may not be one visit, may have to come in and get your one infusion and then come a week later and get your next infusion. But I think the idea of making this entirely outside the hospital is the goal.
Okay. Next one is from Ishtiaq Ahmed.
Which cells survived the virus and what CRISPR target enabled survival? Not quite sure what you're getting at. So if you're talking about bacteria surviving the infection by the virus, I'm not enough of a bacteriologist to know why some bacteria survive and some don't. So I'm not going to guess. I imagine there's other mechanisms that bacteria have to resist viruses. And there's probably some degree of luck and infectious dose and infectious load. In terms of our system, we have found that the virus we use, the AAV6, is so stealthy that basically 90% of our cells survive the viral infection because the virus has evaded the ability of the cells to detect it.
Okay. Fair enough. Next question, please. From Afsan Ashu.
Yeah. Good question. So why does the guide RNA complex does not cut the bacterial CRISPR array? So that's a great question. And I think if the virus, why doesn't it go back and cut the bacterial CRISPR array? Okay, really, really great question and really important. There's a subtle, now we're going to get into the weeds of how the guide RNA Cas9 system works. So the guide RNA, we're going to talk about the strep pyogenes Cas9 because it's the most well understood and the principles are the same for other Cas9s. So there's a 20 nucleotide guide RNA stretch, and adjacent to the guide RNAs is what we call the PAM sequence, the protospacer adjacent motif. And in the incoming virus, there consists both the PAM and the 20 nucleotide guide RNA. But in the bacterial CRISPR array, there isn't the PAM adjacent to the guide RNA sequence. So the guide RNA sequence is, in the bacterial CRISPR array, there's not a PAM, so it can't cut there. But you're right, otherwise it would cut there and you would just be cutting up your own genome. So really, really insightful question.
Next question is from Maisha Farzana.
Yeah, so having worked, are there particular challenges to using this method, or do you think there's a need for an alternative delivery system? I'm a firm believer that that once you have a system, there's always room for improvement. Although we have a saying on my lab chalkboard or whiteboard saying you can't optimize zero, meaning that you need to get a signal first and then you can start improving on it. So I think AAV is a great place to start, but I think there is room for improvement. You'd like to have simpler ways of manufacturing it. While I said I sort of sold it as something where the cells don't detect AAV. Well, the reality is there's a small degree of detection, and we'd like to eliminate that. So I think there might be alternative delivery systems. And I think particularly when we think about trying to do genome editing in vivo, where AAV, many of us, about 50% of us, 50% of us already have an immune response to AAV. I think when we're going to do in vivo editing, we're going to have to develop alternative delivery systems.
Next question from Tanjim Ishaq-Rahman.
Tanjim, you need to read one of we published a paper on this because we had exactly the same hypothesis. So again, let me step back for everyone else. So the most active Cas9 proteins that we use are derived, as Tanjim said, from strep pyogenes, which is a bacteria that causes strep throat and colonizes many of us, and Staph aureus, which is another bacteria that causes human infections, but also colonizes our skin and nose. And because we're exposed to these bacteria, is it possible that from those exposures, we've developed an immune response to the Cas9 that's present in strep pyogenes and Staph aureus? So we went and looked. A really talented technician in the lab, who's now a graduate student, looked. And what he found is about 70% of healthy adults in California have antibodies that recognize strep pyogenes and Staph aureus, and about 50% have detectable T cells. So the answer is yes, they are immunogenic. Well, let me put it this way. Many of us already have an immune response to Cas9. And then if we gave Cas9, even to somebody who didn't have an immune response, because they are foreign proteins, we would develop an immune response. So the ability to redose Cas9 genome editing therapies into humans is going to be a real challenge, which is why we have to think when we're doing in vivo editing, you have to get it all done with the first dose. Now, the advantage for us is ex vivo, we don't expose the patient to any Cas9. By the time the cells go into the patient, the Cas9 is gone. So we don't have to worry about that, which is one of the advantages of first starting with ex vivo cell therapies.
Okay. Next is from Nusrat Afnamika.
Yeah. What can that's a my lab has not spent a lot of time trying to improve the binding specificity or the best performance of the PAM. There's a number of other labs that are doing that. We have used different types of Cas9 proteins that have been engineered to have sort of a better on to off target ratio. And we like the one that was developed by IDT because it maintained the on-target activity. What we found in many of the so-called increased specificity Cas9s is they were almost over-engineered. So they were very specific, but they kind of were too crippled, and they didn't work for us in our system that we use. So you kind of, I don't know if you know the story of Goldilocks and the Three Bears, but the story is that you need to get it just right. If it's too much, it's too much, and if it's too little, it's too little. You need to have it just right. And so this idea of engineering Cas9, you need to hit the sweet spot. I think in terms of the guide RNAs, I think people are starting to look at putting different modifications into the guide RNAs, shortening the guide RNAs. And so there's a number of different approaches. But actually what I'm impressed with is how specific the system is just out of the box. It is an incredibly specific system. And actually, I think it's probably more specific. Sometimes I say it's more specific than life itself. All of us sitting here breathing oxygen, our cells are undergoing mutations all the time. And so what we have to think about is in serious disease, is it worth the small number of off targets? Anyway, great question, Nusrat.
Next is from Nafisa Shoili.
Which one has been read? Well, obviously, I'm in the CRISPR therapeutic space, and I think the potential to revolutionize that is going to happen in the next years. How long it'll take, I'm not sure, but I think it absolutely is going to revolutionize the therapeutic space. I think the diagnostic space is not my specific area, but I suspect it will as well, because of the specificity that it can recognize nucleic acids and pull out nucleic acids. We're going to see a lot of exciting things there too. So both, but I'm particularly excited about the therapeutic space. Thank you.
Next is from Asab Rehman.
Yeah. So, it's not. AAV6 works so well in hematopoietic stem regenerative cells, not because we knew it. It's just because we screened all sorts of other delivery mechanisms. We've tried using some different nanoparticles, and they just don't work as well. We've tried other AAV serotypes, and they don't work as well. So one of the interests in the lab is actually to try to understand mechanistically, why does AAV6 work so well? What is it about this virus, this not even this virus, what is it about this specific serotype of this virus that allows it to work so well? So all I can tell you, Asab, is that it does, and we're basing it on experimental results rather than some, oh, we were super smart and figured this out. The experiments told us.
Next is from Shahimran.
Yeah. So obviously, we want to have high frequencies of the on-target editing that we want, but we don't want to cause off-target edits that might cause harm. And so this balance between on and off-target is really important. And so we'd like to minimize those off-target effects as much as possible. And there's a number of different ways one can do that. I discussed earlier about using Cas9 proteins that are engineered to have a better profile. The other actually advantage of the RMP system is that the Cas9 protein is only delivered as a protein. So it's only in the cell for a very short period of time, probably less than 48 hours. So it doesn't have time to cause a lot of off-target effects. Whereas other delivery systems that have Cas9 expressed for days or weeks at a time, you have more opportunity for off-target effects. So using better proteins, shortening the duration of expression, and then as we talked about, maybe some changes in the guide RNA. But the first two about really just keeping the limit of expression to as short as possible and using the best quality reagents is right now the way we reduce off-target effects.
Next on, we have Tahani Dabasu with a question.
Yeah, so the question is does gene editing cause any adverse effects on regulatory processes such as splicing or the spatial control promoters and enhancers? We don't see that unless, of course, if you intentionally make an insertion and deletion in an enhancer or in a splice site, then of course you've intentionally disrupted it. But in terms of unintentional effects on splicing or silencing of promoters or turning on enhancers, I think that you're getting at, we amazingly don't see that. And again, I don't know why, but we don't. So it's been very nice. Of course, we always worry about that. Have we done something to the property of the cell in the realm that you're discussing? But so far, we don't find that.
Okay. Tayab moving with a question.
Yeah. So AAV6 is considered to be better than AAV2. Would you please clarify is used in most Yeah. So I think so. AAV2 was really the first AAV serotype that was developed. I think it was probably almost 30 years ago now, and for 20 years, maybe not. For 15 years, it was really the only serotype that people use, and AAV2 works very well in many of the cell lines that we use in the lab, and it transduces liver cells. But once people started to knock up against the limits of AAV2, people started looking at these other serotypes. And what we know is that each serotype has properties that lend itself to different applications. For example, AAV9 is the most common serotype because it's able to cross the blood-brain barrier in vivo, which is why the people who are interested in using AAV in vivo for brain diseases are using AAV9. And I think it was really about 5 years ago, my lab, Paula Cannon's lab, Luigi Naldini's lab, sort of all sort of discovered that of the known AAV serotypes, 6 is the best for ex vivo modification of stem cells and other human cells outside the body for ex vivo therapy. So I think that's why AAV2 has historically been most used, but I think as we move forward, everyone's going to start using the serotype that's best for their purposes.
Next on, we have Tanji Mishrak Rahman with a question.
You think creating artificial lasers that help detect. Wow, it's a great idea. Uh, uh, um, I think the challenge is that, the Cas9 is a foreign protein, and so you'd have to somehow develop tolerance to Cas9, and then you'd have to know how to keep it expressed and then have it. It's an interesting idea, but I think there would be a lot of work to be done to really take the bacterial adaptive immune system and really import it into the human immune system. Human immune system does pretty darn well. Let's put it that way. Although it could be, if we got really good at this, one could say, all right, our immune system's never seen this novel coronavirus. Maybe we can enhance the immune system in some way other than a vaccine. But that sounds like a good career-long project.
Next on, we have Sabira Akhtar.
Yeah. So, really, CRISPR's transport systems are really, there's nothing new about these transport systems compared to other drug delivery systems. So I wouldn't say it's any more specific or less specific, because really people are just harnessing what others have developed for drug delivery or gene delivery. So I would say they're about the same.
Next on, we have Maisha Farzana again.
Yeah, thank you, Maisha, for reading our paper with Rosa. Yeah, Rosa and I have a lot of discussions about whether this partial restoration is sufficient or not. And this goes back to the question about whether we should really be targeting exon 1, or should we leave intron 1 intact where there's a lot of regulatory regions and target either exon 2 or exon 3. And so what we're working on now is whether targeting exon 2 or exon 3 gives us better expression than targeting exon 1. That's a first step. Yeah.
Next on, we have Afsan Ashwa.
Yes, I would be happy to.
Taya Moin.
You guys have read my papers. I'm really, so what Taya was referring to is actually a graduate student from Thailand was in my lab, and she showed that she could take MSCs and use target wound healing growth factors into the MSCs, and then use these what we call super potent MSCs, put them into wounds and have them accelerate wound healing. So I think there's a real strong possibility of these working. I think there's the possibility, though, that the growth factors we use are really just maybe version 1.0, and I think we can combine different growth factors and different combinations of growth factors to get even a better effect. But I'd love to see this MSC-based therapy be tested in human patients.
Asaf Rahman with a question.
What is the role of indel as a biomarker in case of it? Yeah, it's Asaf, that's a really great and subtle question. So I think what you're getting at is that there's two types of indels we could use as a biomarker. One is an indel on the target site itself. So LTHSCs, you might have one corrected allele and an indel on the other allele. Can you use the indel on another allele as a signature of that cell? And then, of course, the other indel is the off-target indel that we see. But it's that off-target indel is so infrequent that I don't think we can use it as a biomarker. And unfortunately, the spectrum of indels that occur is not diverse enough, I think, to use it as a true biomarker. What I mean is let's say that we have 1,000 HSCs that engraft. The the indel spectrum is only about 5 to 10. So you would never know if a cell had a certain indel, whether that all the progeny came from that cell or represented 50 different HSCs all with the same indel as a biomarker. I think, we would love someday, we've done this in the lab, and actually the Maldini lab did this as well, is not use the indel as a biomarker, but put into our gene targeting construct a barcode, so that we can create enough diversity to mark each cell uniquely with a barcode, and that would allow us to track. Maybe one day we'll be able to do that in humans, but right now we haven't tried to convince the FDA that would be okay. But it's a very interesting way of understanding stem cell engraftment in biology.
Okay, Maisha Farzana.
Yeah. How would you tackle the emergence of antibodies specific for the transgene product in an IPEX patient body? Yeah, let me Maisha, great question. Let me just step back a bit. I think what you're getting at is that in certain genetic diseases, there's no expression of the protein. And so now if you correct the gene and there's expression of the protein, would the human immune system now recognize this protein as being foreign and develop an immune response to it? So it could be for IPEX, but it could be for many other, that the principle is there for many other patients as well. Now, it turns out in IPEX that most of the mutations aren't what we call nulls, right? Most patient mutations, there's a small amount of protein that's made, or protein is made that doesn't work very well. And so the immune system knows that the protein exists. So in our system for IPEX, really what we would do is just restore functional expression. And I don't think the immune system would react because it already knows the protein's there. But I think in diseases in which there is no protein there and we add back a new protein, then the issue of an immune response will be there. And I think we'll have to think about how do you generate tolerance to that new transgene.
Next from Tayyab Moin.
Is it possible to treat injuries with engineered therapeutic MSCs without replacing the recipient tissue? Yeah, so I think that's what we're trying to get at here, Tayyab, is the cells that we give aren't going to regenerate the tissue, but the cells that we give, like the MSCs, will stimulate regeneration from the endogenous tissue. So we're not replacing the recipient tissue. Instead, we're trying to stimulate the recipient tissue to heal itself better. Because I think, so in this case, when we're doing tissue regeneration using MSCs, we're trying to get the endogenous tissue to regenerate. Whereas in the bone marrow transplant, we are truly doing replacement therapy, where we're trying to replace one set of HSCs with another set of HSCs.
Next on, we have Shagrik Laria.
Yeah, there are now, I think, tens of different Cas9s other than from Streptococcus pyogenes. Some of them have been artificially engineered Cas9s, and there's an ongoing mining. A number of different labs and programs are trying to mine the bacterial space for novel versions of Cas9. And what's interesting is, for reasons that I don't understand, they, like Neisseria meningitidis and Streptococcus thermophilus and so on and so forth. There's many, many out there. None of them seem to work as well as Strep pyogenes, which is really kind of remarkable because I reflect back that Emmanuelle Charpentier was studying Strep pyogenes because that's a human pathogen. If she had been studying some other bacteria and the Cas9 hadn't worked as well, I wonder what would have happened to the field. But this is still the best one, but maybe in the future we're going to get there. And there's clearly other Cas9s that are, it feels like every couple of weeks there's a new Cas9 described. So we're going to have more tools in our toolbox. We already have a lot of tools in our toolbox, and there's just going to be more in the future.
Zuairia Bhakt with a question.
Yeah, so It sounds like graft versus host disease is a major cause of morbidity. Yes, in allogeneic hematopoietic stem cell transplant, graft versus host disease is a really important problem. So I think there's two ways of solving it. One is for genetic diseases, we won't do allogeneic, we'll do autologous, and then there won't be any graft versus host disease. I think when we need to use allogeneic transplantation for malignancies, then I think cell-based therapies are going to be one way to reduce graft versus host disease, including graft manipulation, and trying to get rid of the graft versus host disease causing T cells and either add them back in a non-graft versus host disease way or add back regulatory T cells that prevent it. So there's no one way to prevent graft versus host disease for allogeneic stem cell transplant, which is why I love autologous stem cell transplant, because we don't have to worry about that. Thank you.
Tayab Moin.
Yes. Yeah. So Tayab's asking about the dosage of an MSC-based treatment, and what he's getting at is if you're going to do multiple therapeutic proteins and you're going to mix and match, could you lead to the unwanted proliferation of cells if you don't get the right dosage? And that's exactly right. We need to do a lot of really careful studies to make sure that you have the right growth factors and the right combination and the right mixtures of cells, so you don't get an unwanted proliferation of cells with the wrong mixture. And that's the really careful process that you have to go through as you move from something in the lab to something that you might use in patients. Absolutely.
Thank you. So the next question.
By the way, I love everyone's pictures that show up with their question. It's great.
The next question.
I'm not seeing that.
Okay, I think. Can you see the question?
I only seen Tayeb's last question.
Yeah, this. Okay.
Now I can. I can see Maisha's question. Thank you. Yeah, what steps are necessary to making these approaches more affordable and accessible? Whoops, maybe go back. Yeah, I think several. Well, first of all, probably the most important thing is for people to care and put in the work to make them affordable and accessible. And I think that's true, that there are a lot of people who do care, and we'll as a community will make this happen. What are some of the things that need to happen that are semi-obvious? One is to make the reagents cheaper. Right now, they're very specialized reagents, and so they cost anything that's very specialized initially costs more money. But I think as we use these reagents more and more, they'll become cheaper and cheaper. And then the second thing is that I think we need to turn a manufacturing process for ex vivo therapy into something that, instead of having to be done in a fancy building with a lot of expensive sort of controls and lots of personnel, is to turn it into a process that can be done automated in a device. Now, other people have proposed that the way to make this affordable and accessible is to get rid of ex vivo editing entirely and figure out how to do in vivo editing. And obviously, you can see the appeal. One can see the appeal of in vivo editing, but I think in my talk and in the questions, I think everyone's understood that there's a lot of challenges in vivo editing, like how do you deliver the editing reagents? How do you avoid the immune response? we didn't talk about it yet, but how do you design a delivery reagent that targets the hematopoietic stem cell and not other cells? So I think in vivo editing one day is a possibility, but I think even with ex vivo editing, by reducing the cost of the reagents and by automating the process, I think we can make this more affordable and accessible.
Next on, we have Asab Rahman.
What are the clinical benefit? Yeah. So the preclinical benefits of using the humanized mouse models for evaluating the safety and efficacy of human CRISPR-Cas9 HSC gene on targeting, the great thing about the gene editing system is it's so specific that if you design reagents to target a human gene, they almost never work on the mouse gene. So to really evaluate human reagents, you have to study human cells. And so the benefits of using the humanized mouse models is it allows you to study your human reagents in human cells. The downsides of these humanized mouse models is that, of course, they're mice, and they have entirely different lifespans and physiologies, and even some of the growth factors that regulate hematopoietic stem cells, for example, stem cell factor. There are differences between mouse stem cell factor and human stem cell factor. There's differences in the amounts of these proteins that are in. So they are of great benefit because they're of some use, but they're also limited as well. And I don't think will entirely predict how safe and effective human CRISPR editing will be in humans. And that's why we start with what we call phase 1 trials, which are tests of the safety of the process first in a small number of patients. And that you do the best you can using the humanized mouse models. You compile the data that you can, but in the end, you have to say, okay, this is a profile that looks good enough to move on, but you will never know for sure until you do the human phase 1 trial.
Next on, we have Vishwajit Sarkar.
Yeah, how can CRISPR-Cas9 technology be applied to develop chemotherapy resistance in cancer? Well, we don't want to develop chemotherapy resistance in cancer. We want to prevent chemotherapy resistance in cancer to make our drugs more effective. So, yeah, that's a good question. I think what my thought around how we treat cancers is that cancers will always try to escape. If you give them one thing, they will try to escape one. And so the most effective ways of treating cancer, and actually a similar philosophy or strategy applies to treating HIV, is you have to hit it multiple ways at once to make it very hard for it to escape. And I think that the way the CRISPR-Cas9 technology can work is by developing another set of approaches that hit the cancer. So now you have one more sort of approach the cancer would have to escape, making it even more challenging for it to escape. So I think the CRISPR-Cas9 technology will allow us to develop CAR T cell therapies and NK cell therapies that attack the cancer in a different way than standard chemotherapy. And so if the cancer tries to escape the standard chemotherapy, it hits up against the CAR T therapy. And if it tries to escape the CAR T therapy, it hits up against the chemotherapy.
We have Ishtiaq Ahmed.
Yeah, so that's a great question. So where I draw the line, and I'm only one of many voices. So I believe that human embryos that are after in vitro fertilization, there's often several embryos that are frozen away and would no longer be used. And so the couple has decided they've had enough children and they don't want to do any more IVF. So I think embryos that have would then be discarded, and it's actually sort of troubling to think that they are discarded, but they are. I think they should be released for studying early human development. But right now, I believe that, that after they are after a week, or I think 14 days is the limit, and I think that's a good limit. After 14 days, then the human embryos should not be allowed to develop. But this was going to give us a really great way of understanding early human development that we can't study by studying mice. But I do believe that with current technologies, there's absolutely should be a line that creating pregnancies from human embryos should not be done. The need is not there, and the technology is not close to think about editing live human embryos to create pregnancies.
Next on, we have Maisha Samiha Mahmood.
Yeah. So the reason mRNA is better than plasmid DNA is, and I actually believe protein's even better than mRNA, is for the reason I mentioned in my talk, which is plasmid DNA, when you deliver these things to cells, to get to the nucleus, it has to get through the cytoplasm. And our cells have a robust sensing mechanism for cytoplasmic DNA. TLR9 is a specific example of a sensor of cytoplasmic DNA. And when it senses cytoplasmic DNA, it turns on type 1 interferon and really shuts the cell down. mRNA, which does exist in the cytoplasm, induces that response much less than plasmid DNA, really significantly less, although it still does it. And that's why we like the protein, because the protein doesn't induce this type 1 interferon response at all. But mRNA is clearly better than plasmid DNA.
Next, we have Anika Rayana.
Yeah. Cas9 is derived from 2 common bacterial species. What if the host has developed? Yeah. So it means that it's basically what we're saying is that we've essentially been immunized against Cas9 already. Say 50% of us or more adults have already been immunized to Cas9, which means that delivering Cas9 to that person is going to be very challenging. They're going to will have a hard time getting past that pre-existing immune response. It also means that if we do ex vivo editing, we have to make sure that almost all of the Cas9 is gone before we infuse the cells, because we wouldn't want the immune system to eliminate the cells we've gone to such great lengths to make. So I think this is really an argument, and pushing, I think it goes to an earlier question, is really a drive to identify really good Cas9 systems from bacterial species that don't normally infect humans, and really look to bacteria, as I said, that don't affect humans, and really find the Cas9 systems that work. They're out there. The challenge has been is that often these systems don't work well in mammalian cells. So we need to find something that is, from a exotic bacterial species that also works well in human cells. But I think it'll be found.
Next, we have Maisha Farzana.
Yeah, you guys, I'm impressed. I think you've probably read more of my papers than my lab has read. Why? So we published a paper showing we could edit neural stem cells, and we could put them in a mouse, and the neural stem cells would migrate to different places. And the question is why the migration of the neural stem cells from the subventricular zone to the olfactory bulb? Actually, that goes back to my graduate school work. So there's a natural migration along, I forget the name of it, but there's a tract and a natural migration of cells out to the olfactory bulb in mice. It's not a common migratory tract for humans, but it's present in mice. And so I think what this is showing is that our edited neural stem cells are behaving much the same as the natural cells in the body in migrating along the natural tracts that occur, including out to the olfactory bulb.
Thank you. Next on, we have Shagurika Shah here.
Yeah. Can the CRISPR tool be used to fix frameshift mutations? Absolutely. And you can do it in two different ways. One is use the recombination pathway, which I described, and just fix, insert, insert or delete using recombination to restore the reading frame. But other people have actually used the idea of creating indel frame shift to compensate for the natural frame shift. Now, that won't always work, but if you are in a cell type that is hard to get homologous recombination to work in, it's a clever way of thinking about restoring frameshift mutations by creating a compensatory frameshift. But of course, I like recombination to directly fix the frameshift mutation. Great question.
Thank you, Shakurika. Next question. From Nusrat Afinonomik.
Gene is spectral parent. Yeah. Yep. But these are. It's a great question. So personally, one of the criteria that I listed that our committee said is that this should only be used for conditions that are serious and life-threatening. And I think what you're talking is this vision of using them for traits that aren't serious and life-threatening. And so personally, I would say that that's not something we should do. That crossing the boundary of editing the human doing what we call heritable editing shouldn't be crossed to just modify traits. In terms of who regulates, that is a really complicated question in this era of, of course, genome editing crosses international boundaries. And of course, humans can migrate. Nonetheless, we still live in a world in which nations regulate what goes on. Now, the most recent National Academy proposed that an international regulatory body is convened that has representatives from people all over the world, and that they might serve as the as a committee that every nation could refer to in terms of deciding what would be permissible or not. It's something that's, I think it's a reasonable proposal, and it would allow countries to say, okay, we're not going, you can only proceed with this if you can pass the stringency that this international committee would provide. But that's just one idea, and we'll see. But for now, I think the idea of doing heritable editing to, in a multigenic fashion, to modify traits that aren't associated with serious disease is something we shouldn't do. The other thing that I'm going to even say is that in the U.S. at least, heritable editing or germline editing is presented as a way of creating homogeneity in what people are. And I think the diversity of humans is fantastic. If we were all the same, this would be an incredibly boring world. In fact, it'd be an incredibly unproductive world, right? The fact that we all see the world in a slightly different place, that we all come at the world in a slightly different way, is what makes this world great. And if we think about using genome editing to decrease the diversity, that would be problematic.
Next question from Asap Rahman.
Yeah. How is CRISPR-Cas9 more efficient in mirroring cancer modeling compared to genetic engineered transgenes or homologous recombination in human embryonic stem cells? I think it's just because a lot of the cancer cell lines that we've used don't activate this type 1 interferon response when we use plasmid DNA transfections. And so they are just easier to culture and manipulate and genetically engineer than when we do primary human stem cells. The tools are better. We know how to grow them. They're really robust. They've evolved over decades. But that also means that they have some limitations because they're sort of so artificial. They might not really mimic what's going on in human cancers.
Next question.
What are the challenges of using CRISPR-Cas9 in the treatment of cystic fibrosis? So cystic fibrosis is another genetic disease. It's caused by a mutation in a gene called CFTR, and that causes decreased chloride transport and causes thick mucus in the lungs and the pancreas and the testes and the intestine. So, and the most devastating effect is in the lungs. So I think from a genetic standpoint, fixing the mutations that cause cystic fibrosis is no different than any other genetic disease. The real challenge here is we have now a disease that's not part of the blood system, but is part of the epithelial lining of organs that we don't know how to do ex vivo transplantation for. We don't know how to remove the cells and give them back. And then, well, if we don't know how to do that, well, how about in vivo? Well, the gene therapy that's been used for cystic fibrosis pre-CRISPR had a hard time delivering the transgene to the epithelial cells that needed to be delivered to. So I think for an ex vivo approach, the challenge is how do we get cellular transplantation to work for cystic fibrosis in the lungs and the gut and the pancreas and all the other organs. For in vivo, it's how do we develop genome editing reagents that can actually deliver to the stem cells of the lung and gut and pancreas and so on. And both of those are, I'm glad that both approaches are being worked on because I don't know which one's going to work. And just as I am a firm believer in diversity, I'm also a firm believer that when you don't know what's going to work, you should have multiple approaches working simultaneously.
Next we have Zora.
Yeah. So does the CRISPR-Cas9 system have any potential to help in the ongoing pandemic, for example, in treatment or diagnosis? I think there's my colleagues and there's people from all around the world who have developed sort of proof of concept about how CRISPR-Cas9 might help in diagnostics or in treatment. But I think sort of the more established technologies like PCR and ELISAs and vaccines we're all ready to go. So they will be our solutions to this pandemic. I think, unfortunately, I don't think this is going to be the last pandemic our species has to deal with because there's the exchange of viruses from the environment to the human race is just occurring more and more frequently. So it's possible, though, I hope we never have a next pandemic, but if we do, I think it's more likely when the next pandemic hit that some of these CRISPR tools will be much more mature and ready to help then. I'm not sure they're going to really make an impact on this pandemic.
Thanks, Zora, for the question.
Yeah, very good question.
Next on, we have Andalip, Janlat Ara.
How do you define and quantify genome editing efficiency? Yeah, that's a really important thing to do. And so we use, specifically, we use a technique called digital droplet PCR to quantify our genome editing. But we also use Sanger sequencing of our on-target site and then use for a while, we were using a program called TIDE that was developed by investigators in Holland and the Netherlands. Now we use a program called ICE that is publicly available from Synthago, where you can just load your sequencing trace into the program and it will spit out what percent indels, what percent HR. And then there's advanced things like TIDER and ICER. So we use a combination of ddPCR and Sanger-based sequencing as well. And then finally, we also do some next generation deep sequencing as well, particularly when we're trying to measure off-target indels that are very low frequencies and below the limit of detection of either ddPCR or the Sanger-based methods.
Rian Al-Islam Rashad.
Yeah.
The question.
So hematopoietic stem cells are known to lose their long-term repopulating capacity upon culturing. How can this issue be dealt with? So two ways. One is we try to keep them in culture as short as possible. But as I described, process does involve several days, and it's something that we worry about. And it's something that so first of all, we try to minimize the time in culture. And the second thing we do is there are a lot of groups out there that are developing small molecules or other approaches that allow the stem cell potential to be maintained in culture. And we've been using one of those in ours. We use the small molecule called UM171. And we believe that it is helping maintain the stem cell potential in these. Our culture is about 4 to 5 days. But it's something that we worry about. And luckily, there's so much interest in maintaining hematopoietic stem cell potential that there's new things being reported every day.
Maisha Fonsama.
Yeah. So why is high insertional mutagenesis particularly associated with lentiviral-mediated gene transfer? So really, the appeal of gene editing is we're targeting exactly where we want. Lentiviral gene transfer is an incredibly effective way of delivering new genes into the genome, but it's harvesting an approach in which the virus doesn't have any predisposition to go to one location. Instead, it has the biologic property of integrating into genes that are being used, that are being expressed. And so because the virus has the biologic property of integrating into genes being expressed, it also then has the property that it can cause mutations and inactivate those genes because it's integrating into those, because of its insertional sites, integration sites. So it's really, I think, an almost unavoidable consequence of lentiviral-mediated gene transfer. I will say that we're all very happy in the gene therapy field that while insertional mutagenesis is a property of lentiviral gene transfer, we have not yet seen in human patients any cancer come from that or any leukemia. So all our fingers are crossed that that continues to be the case.
Sabir Akhtar with a question.
Yeah. So what Sabra's question, I think, relates, why is it advised not to create a new sequence when you're doing what I call heritable editing? What could be the consequences of that? And the reason I think somatic cell editing, I think it's okay to create new sequences, just like we talked about with lentiviral vectors for somatic cell therapy. You're using them to treat a serious disease. The changes are not going to be passed along to future generations. It's just going to help that single patient. But when we're talking about heritable editing, what we're talking about is introducing changes that would be passed on along to future generations and perhaps even permeate throughout the human population. And if we don't, you're getting at all my core ethical philosophy. So in addition to believing in diversity, I also believe in humility. And if we're not humble in recognizing that we don't know if a sequence would cause unintended consequences if it was introduced into the human population, then we should be very reluctant to introduce that into the human population. And I think, I won't even call it experiment. The activity done by the person in China showed a lack of humility. He assumed he was smarter than human evolution and that he could create changes in the genome that would be safe. And I think that lack of humility is something that we need to be very careful and avoid, because we don't know whether the sequence changes we introduce, which we may think is right, but we don't know that. And so I think, again, humble.
Raihan Nareed with a question.
Yeah, thank you. That paper just came out. And really was led by my close collaborator, Sergio Pasca, and a fantastic graduate student, Themasab Khan, I think is from Pakistan. Um, and they really use the CRISPR system as a way of modeling 22q11 deletion syndrome. I think, until we have a better understanding, thank you, until we have a better understanding of the genetic basis of neuropsychiatric diseases, and I think we have to be careful about just proposing that, 22q11 is pretty obvious, and if we could somehow restore that deletion, that would be great. But for a lot of other neuropsychiatric diseases, the effect of gene variants is a lot more subtle, and I think we have to have a lot more knowledge about how those variants may cause one thing but be beneficial in another arena before we think about using the CRISPR-Cas9 system. Obviously, there are devastating neuropsychiatric diseases, and that one day we want to develop better therapies for them. So I support the idea, but this is a field I think we need more technical development as well as biologic understanding.
Next from Vishwajit.
Yeah, thank you. That's a good question. So I used to not think about cells as drugs. But as we've been going through the process of developing a gene editing process to be evaluated by the FDA, they don't see, when you give cells to somebody, they don't see them as cells, they see them as drugs. So what we're talking about here is that cell therapies, when you use genome editing of a cell, the product is still a drug. It just, instead of a drug that's a small molecule or an antibody or an enzyme, the drug is the population of cells. So it's really just thinking more broadly about what it means to call something a drug. Hopefully that answers your question. How efficient is engineered MSC in enhancing development of future treatment for chronic diseases? I don't know yet. I hope so. So far, MSC-based therapies have not been very effective for a lot of different reasons. I think MSCs have tremendous potential. But we need more careful study of how they're used, for what diseases are they given, how are the cells purified, how are they cultured, how are they stored. I think in some senses, MSC-based therapy, while it has not caused harm in people, is sort of also been a little bit like the early days of gene therapy, where everyone thought it would be so simple, you just go straight to it and it would all work. I think MSC therapy is really exciting, but probably we need to go back and just be a little more careful about how we use them and how we think about it, instead of assuming that it just will work and it'll be super easy.
Mm-hmm. Next, we have Maisha Samia Mahmood.
Yeah. So the question is, how has gene editing been used to make CAR T cells, quote, universal, and how is it being applied as a treatment for leukemia patients? It's a great question. So currently CAR T therapies, the approved CAR T therapies are autologous therapies. So every patient has their own T cells removed, they're genetically engineered and given back. And that autologous manufacturing process is still quite expensive. Now, as I mentioned earlier, I think there's ways of making it cheaper and more efficient, but for now, it's quite expensive. The other thing is it takes some time. And while you're harvesting the T cells and manufacturing the T cells, the patient their leukemia can progress. And so there are two goals with making universal, or sometimes known as off-the-shelf CAR T cells. One is so that we can make one batch of universal cells and give it to multiple patients, and that could make the whole process cheaper. And the other is they could be available, and you wouldn't have that risk that the patient's leukemia might get worse while you're waiting for the CAR Ts. So how is genome editing being used? So the main thing the reason we don't use off-the-shelf or allogeneic CAR T cells in CAR T therapies is those T cells have T cell receptors on their surface that would cause severe graft-versus-host disease if we infuse them into a cancer patient. And so the way we prevent that is by using genome editing to knock out the T cell receptor and just have the CAR expressed. So that is one way. People are also trying to use genome editing to knock out genes so that the CAR T, the universal CAR T, doesn't get recognized by the host immune system and be rejected. So there's a lot more, but right now it's being used to knock out the TCR, but I think we're going to see more approaches in the future. All right, it looks like we're getting to the award. Oh, yeah.
Zarin Tasnimra. We have more questions.
Yeah. It's great. This is fantastic. So why some Cas9 variants have higher specificity and some optimal on-target activity? Yeah, so what a lot of them did is they put in 3, 4, 5 different amino acid changes, and they got higher and higher specificity, but they didn't screen against maintaining high on-target activity. So I think what they ended up doing is sort of making the entire protein less active. And so you got a better ratio, but you also sort of crippled the protein in the process. And so the reason we like the protein developed by IDT is only has a single amino acid change. And it was identified by screening for both on-target activity and specificity. So it was screened for on 2 different dimensions. And I think that's the reason, is that some of these variants were just over-engineered for specificity and weren't engineered to maintain on-target activity. Great questions, Aaron.
Next,
well, I think the ethical issue is one obstacle. I think there's technical issues that remain and need to be highlighted, that there are still many technical issues. And then I think the issue of whether, and then I think an education issue. We shouldn't, if we go forward with human germline engineering, it should not be a decision that's made by a small group of people, but it's a decision that should be made by a large group of people. But to do that, we of course have to educate people so they understand the field and the consequences of what we might be saying.
Maisha Farzana.
Yeah, do you think CRISPR-derived therapy for any disorder should be patented? Ooh, another really big question. You know, I have mixed feelings about patenting. Clearly, patenting can be a way that slows the dissemination of therapies to more people. Patent protection can be harmful. On the other hand, at least in the US and Western Europe, patent protection gives companies a motivation to develop novel therapies because it gives them protection to make the investment. So I guess where I stand is that I think CRISPR-derived therapies should have some patent protection for a little while, but that it should expire, and then the therapy should be more broadly disseminated without that protection. I think that's the right balance, because if we didn't have patent protection, it might be hard to get companies to invest the tens of millions of dollars it takes to get a CRISPR therapy developed if they didn't know they could make that money back. So that's my balance right there. But it's a really, of course, it's not related to CRISPR either.
Next, we have Asaf Rehman.
Yeah. What are the applications of CRISPR-Cas9 based H's? Yeah. So in principle, Asaf, CRISPR correction system could work for any congenital blood disease, like beta thalassemia, like severe combined immunodeficiency, like Wiskott-Aldrich syndrome. I think there's hundreds, if not thousands, of genetic diseases of the blood and immune system that this system could be applied to. It kind of relates to the earlier question, though. Right now, the development of a CRISPR-based therapy is still pretty expensive. And so people are reluctant to make investments in diseases that are very rare. But I think as we make the developmental process cheaper, in principle, this CRISPR correction system can be applied to a whole host of genetic diseases of the blood and immune system.
We have Yusuf Nayan.
Yeah, thanks, Joseph. Is there any possibility of getting unexpected mutations due to? Yeah, I mean, there's always, I think what you're getting at is, yes, there's always a risk of off-target indels. And, the way we reduce the risk is, I think, by limiting the duration of the Cas9 protein. So it's not being expressed in the cell for any longer than you need it. The shorter, the better. And by, as we've talked about, by continuing to improve the reagents we use, so it decreases the probability. And then I think the other thing we haven't talked about, though, that's a little bit related is also developing better methods of measuring if off-target changes have occurred, and then assessing whether those off-target changes cause any problem. We all are developing new mutations in our body at every second. Most of those have no consequence, and so we need to have better ways of assessing whether an off-target indel would have consequence or not. Thanks, Yosef.
Next, we have Nafisa Shree.
Yeah. What are the major challenges before moving CRISPR technology in clinical use? Well, it's already started to enter the clinic in the US and Western Europe. The challenges, I think, are, since it's a new technology, it takes a special degree of regulation to make sure it's being developed in the correct way. And that's, I think, been the major challenge, is just developing the reagents, the processes, identifying the right first diseases to treat. If those, and I guess I should be an optimist, when those turn out to be successful, then I think the path to clinical use will become more streamlined and more efficient. But right now, because it's so new, the major barrier is just putting everything in place, the manufacturing process, developing good GMP grade reagents, and then developing a regulatory path.
Thank you, Nafisa Shree, for your question. Next one, we have Shah Imran Hussain.
Yeah. Yeah. If CRISPR is used in the treatment, will the world become dependent on IVF more than natural birth? I sure hope not. I think that would be a terrible world. I think that I think that, again, very few couples, are actually at risk of having a child with a genetic disease. And so 99.9999% of couples, their risk of having a child with a genetic disease is, near zero. And so there's no need. So I don't think the world will become dependent on IVF. Or maybe I'm just being hopeful, because I think that would be terrible.
Okay, so next on, this is probably the last question from Mohiminur Rehman Mahin.
Yeah, thank you. Another great question. So the point is that to correct a disease, you don't need to correct a single cell. You might need to correct many cells. And how can this be achieved using the CRISPR technology? Well, for the diseases of the blood and immune system, the way we do that is we can pull many hundreds of millions of cells outside the body and do ex vivo editing, and then give hundreds of millions of cells back. So one of the solutions is to do ex vivo editing, where you do large populations of cells. Now, in vivo, how do you get large number of cells to be corrected is, again, one of the challenges with in vivo editing. How do you develop reagents that allow you to modify many, many cells in a tissue? And we just don't have that solution yet, but people are working on it.
Okay, I think there are more questions.
Okay.
Few more. So
Yeah.
Can we have the next question, please? Yes, there you go.
Yeah.
From Said Anwar.
Right. Hi, Said. Thank you. Nice glasses. Do you think CRISPR can perform better than ZFN-mediated site-specific insertion to treat alpha thalassemia caused by large deletions? I'm relatively agnostic about whether CRISPR is better or ZFN is better or TALENs are better or the next generation is better. What I do, in our own hands, we have found that the RNP CRISPR system does seem to be a little bit better than the ZFN system in terms of doing genome editing. I think alpha thalassemia caused by large deletions, however, is a really challenging thing to correct. We don't know how to put large insertions back into large deletions yet. So that's going to really one of the next frontiers in genome editing is to think about how you could fix large deletions. Now, I will say we're starting to think about alpha thalassemia, but not by fixing the large deletion, but sort of through not a roundabout, but a more indirect approach about fixing the alpha thalassemia gene indirectly rather than directly.
Okay. Saidan were with another question.
Yeah. So we published along with Paula Cannon a comparison of AAV6 with some AAVs that were reported to have high efficiency homologous recombination in HSCs. And both Paula's lab and my lab were unable to show that these other AAVs, these clade F AAVs, had that property. So we're not following up on that. I mean, let me put it this way: we're not following up on the clade F AAVs. We are studying, more of the biology of AAV6. But yeah, we reported that because there was publications saying that they were good. We couldn't reproduce the results, and of course, being able to publish and show that you can't reproduce somebody else's results is an important way that science moves forward.
Next on Sadrina Mona.
Can CRISPR-mediated induced pluripotent technology be used for the development of regenerative medicine or in the field of personalized medicine? I think I think absolutely yes. And in fact, I think CRISPR gene editing is going to help make these iPS-derived regenerative medicines both more effective and safer. So I think absolutely it's going to probably be really important. Since stem cell therapy has been proposed, can CRISPR be applied to improve the efficiency of this approach? I think in diabetes is a complicated disease. There's the type 1 diabetes that's caused by autoimmunity, and there's the type 2 diabetes that is caused by insulin resistance. And I think, broadly, the CRISPR-Cas9 technology, because it's precise and allows you to improve the potency and property of cells, should be able to improve the efficiency, but I'm not enough of an expert to tell you exactly how. But I much like the IPS question, I think as we understand more of the biology about why treatments don't work, we can use CRISPR-Cas9 as a tool to solve those problems and then develop better therapies.
Next on, we have Narita Sanparuke.
Yeah. Thanks, Narita. AAV vectors have been proposed to be more suitable, but won't their integration in host cells lower the editing efficiency in the future due to previously acquired immunity in the host? Well, it's not so much the integration of AAV in the host cells. It's when we give AAV vectors in vivo, they're given in such large doses that our immune response develops incredibly reactive memory to that AAV vector. And it's really that immune memory that precludes the ability to redose AAV in vivo. Now, in what I've described in terms of using AAV ex vivo, the frequency of AAV integration in HSCs is very low. And by the time our manufacturing process is done, the residual AAV we can't detect it. It doesn't mean it's not there. We just can't detect it. And so we don't think we're going to be sort of re-immunizing patients to AAV when we use it ex vivo. Yeah, I disagree with that study. So a study revealed that Cas9 can induce mutations in the tumor suppressor gene in HPSCs. Won't that pose a serious risk of cancer development? Yeah, I don't actually believe that their data supports that Cas9 induced mutations in a tumor suppressor gene. What I think they showed is that whenever you put a population of cells under pressure, you select for mutations in p53. And what I mean by that is mutations in p53 are occurring all the time spontaneously. And when you are putting pressure on a population of cells for proliferation, like we do during Cas9 screens, we end up selecting for clones that have p53 mutations as well as other mutations. But in our hands, we have had no evidence that Cas9 itself causes those mutations. So I disagree with the conclusion of that paper.
Next, we have Dangerous.
Thank you for the compliment.
Yeah.
Right.
How does our body make?
Yeah. So so the way our body makes specific receptor proteins for microbes is through the process called VDJ recombination, in which we have an array of genes that make our T cell receptor, and we have an array of genes that make our immunoglobulins. And actually, what's really interesting is we create diversity in VDJ recombination through what I call the body's natural genome editing system. So instead of using CRISPR to induce rearrangements to create antibodies and T cell receptors, we use the RAG1, RAG2 nuclease that binds to specific target sites and rearrange these segments of genes as a way of creating antibodies and T cell receptors to fight microbes. So I'm not sure we can enhance that natural recombination process by CRISPR, but I think we might be able to help give immune systems the best antibodies and best T cell receptors using the CRISPR system. But the natural VDJ recombination system is really quite remarkable, and I don't know if we can improve on it. Thank you. Thank you.
Everyone.
And I'm impressed that you guys are all awake at 1 a.m.
Actually, we had more questions, but since it's too late in Bangladesh, we actually have to end the question and answer segment over here. But really amazed to see the participation of the viewers. So thank you everyone for making this segment amazing. Yes, Sadrina over there.
Yeah, I mean, yes. Thank you again for the compliment. My lab is currently full, but I'm always proud of the successes of the people in the lab, and they move on to the next positions. And so there's always, don't have a lot of people come through the lab, but there is those opportunities. So thank you.
Thank you so much. So, okay, so now the most awaiting moment of the CRISPR Feast 2020. Yeah. Throughout the entire event, community of biotechnology arranged for competitions like research idea presentation, poster photography, CRISPR meme, CRISPR debate, CRISPR Olympiad. And I'm very glad to inform all of you that we received incredible responses from the participants. So through the process of intensive selection by our experienced judges, we have come up with a list of the best performers. And now I would like to request Dr. Matthew Porteus to announce the name of the best performers, as well as the most intriguing question asked in this session. So if you could just read out the names, I'm going to share my screen.
Yep.
All right. Can we have the slide, please?
There we are. I'm honored to be able to announce the winners. This is great. And based on the questions, I'm sure there were fantastic winners.
Okay, so
The winner for the research idea presentation comes from Team Fish. What a great name for a team. From Afia Alam, from Brock University, Maisha from Brock University, and Shamran from Brock University. Congratulations to Team Fish.
Congratulations, Team Fish. Next, we have the CRISPR debate.
The winner of the CRISPR debate is Team Cas9, led by two medical doctors. Oh, I'm going to have to help me. Mamanu Zaman from University of Rajshahi, and Ashikar from University of Rajshahi, and Fahima from University of Rajshahi. Congratulations to Team Cas9.
Winners of CRISPR Olympiad.
Yeah, the winner of the CRISPR Olympiad in Group A was Wasima from Sylhet Agricultural University, and the winner in Group D was Nishat from Soest. Congratulations to them. CRISPR means. The second runner-up is Duraya from Brock University, BRAC University, and the first runner-up is Sakalian from Sylhet Agricultural University. Congratulations to both of you. Oh, and the champion, sorry, is Sadia from University of Dhaka, right there in, yeah, congratulations to all three of you. And we have honorable mentions. Oh, must have been a really challenging, must have been a great group. Honorary mention to Mitra Binda from Institute of Applied Medicines and Research in India, an international competition.
Congratulations.
Explaining CRISPR in 2 minutes, the second runner-up is Arif from Maulana Bashani Science and Technology University. Oh, I bet they did a better job than me. I should have listened into this. First runner-up is another MD, Ashikar from University of Rajshahi, and the winner was Wasima from Sylhet Agricultural University, who were able to explain CRISPR in 2 minutes. Congratulations, that's a challenge. Poster photography. The second runner-up is Riyal Chowdhury from University of Science and Technology in Chittagong. The first runner-up is Umamazam from University of Rajshahi, and the winner was Mumtaza from University of Chittagong. Congratulations to all three of you. Oh, and an honorary mention to Eric Moreta from Ecuador. Truly an international competition.
Okay, so now we have to announce the most intriguing question asked in this session. So now you have to announce that.
Oh my goodness, I didn't know that. Okay. Most intriguing. They were. First of all, let me compliment you all on all of the great questions and the thoughtfulness of the questions. They were, below. They weren't just at the surface level. They were very deep questions. I would say, oh man, I can't. You're putting me on the spot. I would say.
I know there were many interesting questions actually.
Yeah.
Like when you were asking, so yeah.
Perhaps the question that will probably the one that will I'll spend the rest of since it's morning here, spend some of my day thinking about more is how do we harness the genome editing system in better or the CRISPR system in better ways to treat cancer. I think it's really a next generation of doing that. There's certainly ideas out there, but I think we need to do more thinking about how do we really harness the CRISPR system to treat cancer.
Yes.
But that's
so thank you so much.
It's so unfair because there were so many good questions, and I hate to pick only one.
Actually, there were more questions, but due to shortage of time, we were not being able to show it. So probably next time.
Maybe next time. Well, congratulations on holding this CRISPR Fest in these incredibly weird times in a virtual setting. But I'm also, while I'm sad that I can't be there to see everyone in person, it's in fact great to be able to participate from around the world and in this.
Thank you so much. Thank you so much.
Yeah.
So, uh, this was actually actually an amazing session, I must say. Hope the viewers could take some notes from today's event. All thanks to Dr. Matthew Porteus for sharing all these amazing ideas with all of us. I would like to give special gratitude to Dr. Matthew Porteus for joining us today amidst his busy schedule. I know you have been you are busy, but still, thank you so much for joining us. Hope to have you again in future.
Thank you for inviting me. It was really an enjoyable way and really fantastic. So congratulations.
Thank you, thank you. I would
Thank you.
also like to thank our advisors who have been our support system throughout the entire event. Thanking Joan Kamens, Francisco Mojica, Jennifer Doudna, Martin Janek, who have sent us best wishes for the event. Also, thanks to all our viewers who have been with us throughout the session. The session would not have been successful without all of your support. Last but not the least, I would like to take a moment to thank the entire team of Community of Biotechnology who have been working really hard for the past few months in order to gift us such an amazing event. So thank you everyone for joining us. See you next time. Till then, stay safe, stay with
Thank you.
COB. Thank you.
Thank you. Stay safe. Bye-bye.